Good afternoon, everyone. I would like to start by thanking the organizers for giving me this opportunity, and I am excited to be telling you today about this new approach that we have developed to analyze three-color microscopy images and learn from them the colocalization relationships and hierarchies between three molecules. To start, colocalization is a standard approach in cell biology and other fields. There are many different flavors of it. There have been many developments over the years, but almost all of them are focused on two molecular entities at a time and asking whether they colocalize with each other or not. We are, however, interested in three molecular entities and in learning the colocalization relationships between them. And so we developed this new algorithm, which I will be telling you about, and this is the work primarily of a graduate student, Vega-Lugo, in the lab. So the question we're asking really is the extent of colocalization between two molecular entities influenced positively or negatively by a third molecular entity? So at a glance, our approach that we developed is object-based, which means that you first detect or segment the objects in the images, and then you perform colocalization analysis on the segmentations and detections. It assesses colocalization using spatial point pattern analysis, and particularly using nearest neighbor distances. It is applicable to both diffraction-limited punctate objects as well as larger non-punctate objects. For the latter, we generalize the definition of nearest neighbor distances, and you can read more about this in our upcoming publication in JCB. So conceptually, what we do is that we divide the three molecular entities into target, reference, and condition. That's what we call them. And the question we are answering is the extent of target colocalization with reference influenced positively or negatively by target and or reference colocalization with condition. And of course, by permutating who is the target, who is the reference, and who is the condition among the molecular entities, one can probe these relationships from all angles. So let's get into a little bit more detail. First, let's look at what measures we've devised to assess the influence of target colocalization with condition on target colocalization with reference. For this, we take our target objects, let's say we have n sub t of them, and we divide them first into colocalized with condition or not. And this is done by comparing their distance to the nearest neighbor condition to a colocalization radius. And I will come back to that later towards the end of my talk. And then we take these two subsets of target and we ask what fraction of them is colocalized also with reference. As you can see here, so this is the subset of target that is colocalized with condition and reference. This is the subset that is not colocalized with condition, but is colocalized with reference. And from these analyses, basically, we can calculate these conditional probabilities of target to colocalize with reference, if target is colocalized with condition, or in this case here, if target is not colocalized with condition. And we can also compare these conditional probabilities to the overall probability of target to colocalize with reference, regardless of what the condition is doing. And by comparing these three probabilities to each other, we can assess what is the influence when target is colocalized with condition. How does that influence target colocalization with reference? We can do a similar analysis, at least similar in spirit, to assess then the effect of condition on reference. So here we're asking how much influence does reference colocalization with condition have on target colocalization with reference? So in a similar fashion, we basically take now the target objects and we divide them now into those with reference and those without. And then for those that are with reference, we look at these reference and we ask how many of them are with condition or not with condition. And from these subdivisions, we can calculate colocalization measures shown here in these equations, which reflect the influence of the reference colocalization with condition on target colocalization with reference. You probably noticed these look a little more complicated than the measures on the previous slide. That's because we have to compensate for the fact that there are different number of reference objects in the total versus with condition versus not with condition. But this rescaling shown here takes care of this and allows us to compare these measures again to the overall probability of target to colocalize with reference to then assess the effect of condition on reference for target reference colocalization. So this is the method in very, very briefly, and we've benchmarked it and we validated it using simulations and using experimental negative and positive controls. I am not going to go into those today. Rather, I'm going to show you one example to both illustrate the method and show you what kind of results one can learn from it. So our application is focused on the cell surface receptor VEGFR2 and its downstream adapter TSAT. And we were interested in basically learning whether the interactions, as reflected by colocalization between VEGFR2 and TSAT, whether they would be enhanced at focal adhesions. Because interestingly, VEGFR2 TSAT signaling leads to focal adhesion turnover. And we noticed that VEGFR2, some of it at least likes to hang out around adhesions. And so we were wondering whether that facilitates VEGFR2 signaling. And so we're going to focus for the rest of my talk on these three molecules, VEGFR2 and TSAT, and pexillin as a marker for focal adhesions. And so to do this, we took 3-color fixed cell immunofluorescence images of the 3 molecular entities, as you can see here. Then we detected the VEGFR2 and TSAT objects in our images, and we segmented the pexillin patches to figure out where focal adhesions are, and then we could perform the conditional colocalization analysis that I described to you. And what did we learn? So let's go through the analysis results step by step. First, if we look at just the overall probability of target to colocalize with reference, and what I'm showing here is VEGFR2 colocalizing with TSAT. Interestingly, we see that overall there is no significant colocalization between VEGFR2 and TSAT. So blue are the data, and in black is basically what we expect for coincidental colocalization. And as you can see, there is no difference between them. And this coincidental colocalization is obtained by replacing the target with basically objects with points on a grid. So no colocalization so far. But when we look at the different subsets, the subset which does stand out is the subset of TSAT that is colocalized with focal adhesions with pexillin. So now this measure is actually significant, as shown here by this pound sign. And how do we assess whether it is significant? We actually do 3 statistical tests. So this is basically significantly greater than the black box, which is the same as the null TR here that I showed before. Greater than also the green box, which is for RAND-C, which I'll come back to in a second, and also greater than the overall probability. So that's why this colocalization measure is significant. So how is this green box, this RAND-C, calculated? Sort of in a similar spirit as what we do here to abolish the target reference relationships. Here we randomize the positions of the condition objects in order to abolish any true influence of the condition on the target and reference. And so now back to this colocalization radius. As you can see here, we're using a colocalization of 3 pixels, which if you think about it, is pretty large. That's about 240 nanometers, which is very large for molecules. But that's more or less what we need for our kind of images, given registration shift, focalization, precision, etc. But actually, what we found out is that if you do the analysis for a range of colocalization radii, that is how you get the most robust results. So as you can see here, so what I'm showing you here is actually what we call a minus log 10 p-value table. So from these comparisons, we take the p-values and basically show the minus log 10 of those p-values. And you can see for the overall, no matter what colocalization radius we use, it's all not significant. But for the subset of TSAT with focal adhesions, between the colocalization radii of 2 and 5, we have pretty significant colocalization values. So a true actually indicator of true relationships is when you have significant colocalization measures over a range of colocalization radii. And I'm not showing you the data here, but we permutated target and reference and condition between these 3 molecular entities. And this analysis revealed that there is cooperativity between VEGFR2 TSAT colocalization and TSAT focal adhesion colocalization. So, and we're trying to figure out now more by looking at more molecules as well as life cell data, what exactly is happening there. So in a nutshell, in summary, we've developed this new approach to analyze the conditional colocalization relationships and hierarchies between 3 molecular entities from 3-color microscopy images. As I mentioned, and as I showed, the approach is applicable to both punctate and non-punctate objects. Analysis over a range of colocalization radii yields the most robust results, and that's what we will be doing moving forward and what we recommend. And this approach is implemented in the MATLAB code, which is freely available. And you can download it from our GitHub page. And thank you for your attention, and I'm happy to answer any questions.